The weight you've been carrying in your body today, that deep, bone-level tiredness, has been quietly asking for something all day. Science has a name for what's happening. When you finally stop, when you finally close your eyes, your body doesn't go dark. It shifts into something more active than most of your waking hours. A repair program so intricate, so precisely coordinated, that engineers have studied it for decades and still don't fully understand it. And here is the part most people miss the more exhausted you are. The more the body needs you to stop fighting and let it work. Forcing more wakefulness when the system is signaling repair doesn't give you more. It costs you the very thing trying to restore you. Welcome to Sleepy Mind Science. Tonight begins where the exhaustion ends. Close your eyes and breathe gently. Let the weight of the day settle. Your body already knows what to do, and it has already started. And now we begin. The human brain accumulates a protein called beta amyloid throughout every waking hour. This protein is a byproduct of normal neural activity, the metabolic waste left behind when neurons fire and communicate. During the day, small amounts build up in the spaces between brain cells, too slowly to cause immediate harm, but persistently enough that by evening, a measurable amount has gathered. Researchers have found that even a single night of poor sleep leads to a significant increase in beta amyloid in the prefrontal cortex and hippocampus, the regions responsible for decision-making and memory. The clearance system that removes beta amyloid is nearly inactive during waking hours. This is where tonight begins, with the question your body has been quietly asking all day. The brain has its own dedicated waste clearance system, discovered only in 2012 by neuroscientist Maiken Nedegard and her team at the University of Rochester. They named it the glymphatic system, a combination of glial cells and the lymphatic system it resembles. Glial cells are the brain's support network, outnumbering neurons by roughly 10 to 1. Nedegard's discovery revealed something unexpected in animal studies. Glial cells shrink by nearly 60% during sleep, opening wide channels between brain cells. Cerebrospinal fluid then flows through those channels like a slow, gentle current, flushing metabolic waste out of the brain and into the broader lymphatic system. Whether the same magnitude of shrinkage occurs in humans is still being studied, but the clearance function itself is well established. The brain, it turns out, cleans itself, but only while you sleep. Cerebrospinal fluid is a clear, colorless liquid that cushions the brain and spinal cord. The body produces about a pint of it every day, cycling it continuously through a sealed system. During waking hours, this fluid primarily performs mechanical functions, absorbing shock, maintaining pressure, delivering a small number of nutrients. During sleep, its role transforms. Imaging studies have shown that cerebrospinal fluid pulses in large, rhythmic waves through the sleeping brain, synchronized with slow electrical oscillations. Each pulse pushes deeper into the brain tissue than anything seen during wakefulness. These waves are not random. They are coordinated with the heart, the breath, and the electrical activity of sleeping neurons, a precisely timed rinse cycle running through the night. The glymphatic system operates most actively during slow-wave sleep, also called deep sleep or non-REM stage 3. This is the sleep stage that dominates the first half of the night, the stage most often lost when people sleep fewer than 7 hours. During slow-wave sleep, the brain's electrical activity slows to large, synchronized waves called delta waves, oscillating at roughly 1 cycle per second. These waves appear to act as a pressure driver, creating differentials in brain tissue that draw cerebrospinal fluid inward and push it outward in a regular rhythm. Researchers are still mapping the precise mechanics, but the coordination between deep sleep and glymphatic clearance is one of the most consistent findings in recent sleep neuroscience. The cleaning isn't passive. It is driven by the sleeping brain's own electrical activity. Among the waste products cleared by the glymphatic system is tau protein, another molecule associated with neurodegeneration. In healthy brains, tau helps stabilize the internal structure of neurons. But when neurons are stressed or damaged, tau can become abnormally modified and begin to tangle. Researchers have found that tau accumulates faster in sleep-deprived brains, and that glymphatic clearance of tau is significantly reduced after even one night of disrupted sleep. Studies using specialized brain scans have shown elevated tau tangles in the brains of people who consistently sleep fewer than 6 hours per night. The connection between sleep, glymphatic clearance, and long-term brain health is one of the most active areas in neuroscience right now. And the findings consistently point the same direction. The glymphatic system does not distribute its cleaning effort equally across the brain. Certain regions receive more clearance than others, and the prioritization appears to follow functional logic. The hippocampus, the brain's memory consolidation hub, receives particularly intensive glymphatic flow during deep sleep. So does the prefrontal cortex, which handles executive function, emotional regulation, and judgment. These are precisely the regions that show the most functional impairment after sleep deprivation, and the regions most heavily burdened with metabolic waste after a long day of cognitive effort. The brain appears to clean hardest where it has worked hardest. The exhausted mind is not passive. It is methodical. Astrocytes are a type of glial cell that plays a central role in the glymphatic system. During waking hours, astrocytes are busy supporting neurons, regulating ion concentrations, recycling neurotransmitters, and maintaining the chemical balance that allows electrical signaling. When sleep begins, astrocytes shift their role dramatically. They contract, pulling their cell bodies inward and opening the perivascular spaces, the narrow channels that run alongside blood vessels throughout the brain. It is through these channels that cerebrospinal fluid flows most freely. The contraction of astrocytes is triggered in part by adenosine, a molecule that accumulates with waking and signals sleep need. The same chemical that makes you feel tired is also the signal that opens the brain's drainage pathways. Adenosine is a molecule the brain produces continuously during wakefulness as a byproduct of cellular energy use. Neurons use ATP, adenosine triphosphate, as their primary fuel, and adenosine is what remains after ATP is consumed. It accumulates in the brain throughout the day, binding to receptors that progressively slow neural activity and generate the sensation of sleepiness. Caffeine works by blocking adenosine receptors. It doesn't reduce adenosine itself, only prevents it from binding. During sleep, adenosine is cleared from the brain, which is why waking after sufficient sleep feels like a reset. Adenosine accumulation also drives the depth of slow-wave sleep, effectively calibrating how intensively the glymphatic system works. More exhaustion, more adenosine, deeper cleaning. The blood-brain barrier is a tightly regulated boundary between the brain's blood supply and its tissue, formed by specialized cells lining the brain's capillaries. It blocks most substances from passing freely into the brain, protecting neural tissue from pathogens and chemical fluctuations in the bloodstream. During sleep, the blood-brain barrier undergoes subtle but meaningful changes. Its permeability shifts in ways that appear to support waste removal, allowing certain metabolic byproducts to pass outward more easily. Researchers have found that chronic sleep deprivation gradually compromises blood-brain barrier integrity, allowing inflammatory molecules to enter brain tissue that would normally be excluded. Sleep is not just a clearance event. It is a maintenance window for one of the body's most precise structural systems. So what does the exhausted body actually do to repair itself while you sleep? The first answer is this: it opens what was closed during waking hours. Drainage channels, clearance pathways, metabolic reset systems, begins to unseal within minutes of sleep onset. The glymphatic system activates. Cerebrospinal fluid begins its slow, rhythmic passage. Adenosine begins to clear. The brain's waste begins to move. None of this is passive waiting. It is an organized, initiated process that requires sleep to begin, and it cannot be replaced by rest, meditation, or stillness while awake. The body knows the difference between lying down and actually sleeping. Tonight, this is what is happening. Beyond the brain, sleep initiates a cascade of repair processes throughout the body that have nothing to do with glymphatic clearance. Human growth hormone, one of the body's primary agents of tissue repair, is released in its largest daily pulse during the first cycle of deep sleep, typically within 90 minutes of sleep onset. This hormone triggers protein synthesis in muscles, facilitates the repair of microscopic tissue damage accumulated during the day, and signals fat cells to release stored energy for cellular maintenance. Its release is tightly linked to slow-wave sleep, not to clock time. Sleeping at irregular hours or getting insufficient deep sleep significantly reduces the nightly growth hormone pulse, slowing the body's repair cycle regardless of total hours in bed. The immune system undergoes a profound reorganization during sleep. T cells, a type of white blood cell central to immune defense, becomes significantly more effective at finding and binding to infected cells during sleep. A 2019 study found that sleep enhances T-cell activity, partly by reducing levels of adrenaline and other stress hormones that suppress immune cell adhesion during waking hours. The drop in cortisol and adrenaline that accompanies deep sleep creates a biological window where the immune system can work more freely. Cytokines, small proteins that coordinate the immune response, are produced in higher quantities during sleep, particularly during fever and active infection. The phrase "sleep it off" reflects a genuinely understood biological process. Muscle repair is one of the most tangible examples of sleep's restorative function. Physical activity, even ordinary daily movement, creates microscopic damage to muscle fibers. This damage is normal and intentional. The inflammatory response it triggers is what drives muscle maintenance and, in the case of exercise, adaptation and growth. But the repair itself happens almost entirely during sleep. Satellite cells, specialized stem cells that live alongside muscle fibers, activate during sleep to patch damaged tissue, Protein synthesis rates in muscle tissue are significantly higher during deep sleep than during waking rest. Studies examining recovery in athletes have found that sleep extension accelerates physical recovery more effectively than most nutritional or pharmacological interventions. The cardiovascular system uses sleep to accomplish maintenance that waking physiology doesn't permit. Blood pressure drops significantly during non-REM sleep, by as much as 10 to 20% in healthy adults, a phenomenon called nocturnal dipping. This pressure reduction lowers the mechanical stress on arterial walls, allowing vascular tissue time to repair. Endothelial cells, the thin layer lining blood vessels, have higher rates of repair and regeneration during sleep. Studies have consistently found that people who do not experience adequate nocturnal dipping, often due to sleep disorders or fragmented sleep, have higher rates of cardiovascular disease, arterial stiffness, and hypertension over time. The heart rests, but the vessel walls quietly rebuild. The liver operates on a circadian rhythm that is tightly synchronized with sleep. During sleep, the liver shifts from active metabolic processing, filtering nutrients from a day's worth of food, to a different mode producing repair proteins, clearing cellular debris from the bloodstream, and synthesizing factors needed for tissue regeneration elsewhere in the body. Disrupted sleep cycles desynchronize the liver's internal clock from the brain's master clock in the suprachiasmatic nucleus, impairing its metabolic function. Studies in shift workers, whose sleep is chronically misaligned with natural light cycles, show elevated rates of liver inflammation and metabolic dysfunction compared to those who sleep in alignment with their circadian rhythm. The body's organs repair on the same schedule. The skin undergoes measurable repair and regeneration during sleep. Skin cells divide more rapidly during the night than during the day, following a circadian pattern driven by cortisol and growth hormone. Collagen synthesis, the process that maintains skin structure, elasticity, and healing capacity, peaks during deep sleep. The skin's barrier function, which protects against moisture loss and environmental damage, is also strengthened during sleep. Studies have shown that sleep deprivation impairs wound healing, slows skin barrier recovery after disruption, and accelerates measurable signs of skin aging over time. The phrase beauty sleep is a simplification of a genuinely documented biological process. Nighttime is when the skin rebuilds what the day erodes. The kidney operates differently during sleep than during waking hours. Filtration rate slows, and the organ focuses less on removing metabolic waste from the bloodstream and more on concentrating urine. One reason most people don't need to urinate in the middle of the night when sleeping well. This shift is regulated by antidiuretic hormone, which rises sharply at sleep onset and suppresses urine production. Sleep deprivation disrupts this hormonal pattern, which is one reason poor sleepers often report waking to urinate and have higher rates of nocturia. The kidney's nighttime behavior is not merely passive conservation. It is an active recalibration of fluid and electrolyte balance that prepares the body's chemical environment for the next waking day. The gut and the sleeping brain are in constant communication through the vagus nerve and a shared hormonal language. During sleep, the digestive system enters a maintenance phase. Intestinal cell turnover increases. The mucus lining the gut wall is renewed, and the populations of gut bacteria shift in ways associated with reduced inflammation. Research in recent years has linked chronic sleep deprivation to altered gut microbiome composition. Specifically, a reduction in bacteria associated with metabolic health and an increase in those linked to inflammation. The gut-brain axis means these microbial shifts are not isolated to digestion changes in gut bacteria during sleep disruption produce measurable downstream effects on mood, cognition, and stress reactivity in the brain. The endocrine system, the network of glands that produces hormones, follows a sleep-dependent release schedule that governs much of the body's repair chemistry. In addition to growth hormone and cortisol, several other hormones are released or suppressed in patterns tied specifically to sleep stages rather than simply to time of day. Prolactin, associated with immune function and tissue repair, rises during sleep. Melatonin, secreted by the pineal gland in response to darkness, coordinates the timing of multiple repair processes across different organ systems. Testosterone, which contributes to muscle repair and cellular maintenance in both sexes, peaks during REM sleep. The sleeping body is not a passive machine awaiting morning. It is a pharmacy running its most complex dispensing schedule of the day. The eye undergoes intensive repair during sleep, specifically during REM sleep, when rapid eye movements reflect intense internal neural activity. The cornea, which has no blood supply and relies on tears and aqueous humor for oxygen and nutrients, benefits from the extended closed-eye period during sleep to allow surface cell repair. The trabecular meshwork, a drainage tissue in the eye that regulates intraocular pressure, shows higher repair activity during sleep. Intraocular pressure follows a circadian rhythm, peaking in the early morning hours and declining through the day. Disruptions to this rhythm, associated with chronic sleep problems, are a recognized risk factor for glaucoma progression. Even the eye, which feels passive during sleep, is actively maintained. The spine decompresses during sleep in a way that is not possible during waking hours. The intervertebral discs, the cartilage-like pads between vertebrae, are compressed by gravity and the weight of the body during the day. They contain a gel-like core that slowly loses fluid under sustained pressure. During sleep, particularly in the early hours, the discs rehydrate as pressure is removed. This rehydration restores disc height and flexibility, which is why most people are measurably taller first thing in the morning than they are at the end of the day. The difference is typically between half an inch and a full inch. The spine rebuilds itself overnight, not metaphorically, but structurally, measurably, precisely. The brain's synaptic connections, the contact points between neurons, undergo a calibration process during sleep described by the synaptic homeostasis hypothesis, developed by researchers Giulio Tononi and Chiara Cirelli. During waking hours, learning and experience strengthen synaptic connections across the brain. If this strengthening continued unchecked, synapses would eventually become saturated, unable to encode new information, requiring more and more energy to maintain, generating increasing levels of neural noise. During deep sleep, the brain selectively weakens some of these connections, pruning the network back to a more efficient baseline. The facts and experiences deemed important are preserved and consolidated. The rest is gently let go. Sleep, in this sense, is not just rest. It is editorial work, the night shift of memory. Memory consolidation during sleep is not a passive replay of what happened during the day. It is an active reconstruction. During slow-wave sleep, the hippocampus, which temporarily stores new memories, replays recent experiences to the cortex at roughly 20 times their real-time speed. The cortex, receiving these replayed fragments, integrates them into existing long-term knowledge structures. This process, called systems consolidation, slowly transfers memories from temporary hippocampal storage to stable cortical storage over the course of nights and weeks. The memories that survive are not necessarily the most recent or the most consciously reviewed. They are the ones most meaningfully connected to what the brain already knows. Sleep doesn't just store the day. It decides what the day means. Emotional memory receives special processing during REM sleep, a stage that occurs in longer, more frequent episodes in the second half of the night. During REM, the brain revisits emotionally significant memories in a neurochemical environment, notably different from waking. Norepinephrine, the brain chemical associated with stress reactivity, is nearly absent during REM sleep. This creates a condition in which emotional memories can be processed and recontextualized without the full physiological stress response they originally triggered. Researcher Matthew Walker at UC Berkeley has described this as the brain's built-in overnight therapy. Experiences that feel sharp and destabilizing in the evening often feel more manageable by morning, not because they were forgotten, but because they were processed in a calmer chemical state. So we arrive at a quarter of the way through tonight's journey, carrying this: the exhausted body is not a broken machine waiting for morning to restart. It is a repair laboratory running one of the most complex and precisely timed protocols in biology. Every system, brain, immune, cardiovascular, musculoskeletal, endocrine, gut, has a sleep-dependent maintenance window, and those windows have already opened. Right now, as your body settles and your breathing slows. These systems are beginning to do what they were designed to do. What does the exhausted body actually do to repair itself while you sleep? It does everything. And it has already started. Let's pause here and notice your body. As you breathe out slowly, appreciate what is happening beneath the surface. Trillions of processes running in silence. Cells dividing, proteins folding, channels opening, all without your instruction. Your body doesn't need you to help. It needs you to stop. Feel the weight of your muscles against the surface beneath you. Notice the gentle rise and fall of your chest. You are a complete repair system, self-organizing, self-correcting, patient. Now we continue. The brain's neural networks reorganize during sleep in ways that produce genuine creative insight. The term "sleep on it" reflects a well-documented phenomenon. Problems that resist solution during waking concentration often yield to novel approaches after sleep. During REM sleep, the brain activates regions associated with associative thinking and loosens the suppression of unusual neural connections that waking logic enforces. A study published in Nature found that participants who slept after learning a mathematical puzzle were 3 times more likely to discover an elegant shortcut solution than those who stayed awake. The repair the sleeping brain performs is not only structural, it is architectural. New pathways form. Old patterns relax. Solutions emerge from a reorganized mind. Sleep architecture. The cycle of sleep stages through the night follows a predictable but personally calibrated pattern. A typical night consists of 4 to 6 sleep cycles, each lasting roughly 90 minutes. Early cycles are dominated by slow-wave deep sleep, when lymphatic clearance and growth hormone release peak. Later cycles contain progressively more REM sleep, the stage associated with emotional processing and creative consolidation. This architecture reflects a deliberate biological prioritization: repair the physical infrastructure first, then process and consolidate experience. Disrupting this sequence, whether by cutting sleep short or by alcohol, which suppresses REM, doesn't simply reduce sleep. It selectively removes stages with specific repair functions, leaving the work half done. The default mode network is a set of brain regions that activate together when the mind is not focused on external tasks. During rest, daydreaming, and self-reflection. It includes the medial prefrontal cortex, the posterior cingulate cortex, and the angular gyrus. During waking mental exhaustion, the default mode network often runs in overdrive, generating repetitive self-referential thinking, replaying concerns, constructing scenarios. This is part of what makes mental fatigue so different from physical fatigue. The brain that feels exhausted is often still running. During deep sleep, default mode network activity quiets dramatically. The regions that generate rumination and self-monitoring settle into a baseline that waking exhaustion rarely allows. Sleep does what willpower cannot. Cortisol, the body's primary stress hormone, follows a strict circadian rhythm with a key function during sleep. Cortisol levels decline sharply in the hours before sleep, reaching their lowest point around midnight to 2 in the morning. This trough is an active, permissive state, a window during which cellular repair processes that cortisol normally suppresses can proceed. The immune system, inflammatory regulation, and tissue regeneration all benefit from this cortisol nadir. In people experiencing chronic stress, cortisol remains elevated into the night, compressing or eliminating this repair window. This is one mechanism by which chronic stress causes physical deterioration, not simply through daytime wear, but by narrowing the nighttime window when the body recovers. The prefrontal cortex is the last brain region to fully develop in adolescence and the first to show functional impairment under sleep deprivation. This region manages executive functions planning, impulse control, emotional regulation, abstract reasoning, and social judgment. After 17 to 19 waking hours, the equivalent of a normal day, prefrontal function begins to measurably degrade. After 24 hours without sleep, prefrontal impairment is comparable to legal alcohol intoxication. Yet sleep-deprived individuals are notably poor at assessing their own impairment, because the prefrontal cortex is also the region responsible for self-evaluation. The most cognitively important region of the brain is the one most sensitive to sleep deprivation and the least able to detect its own compromised state. Neuroplasticity, the brain's ability to reorganize itself by forming new neural connections, is profoundly dependent on sleep. Long-term potentiation, the cellular mechanism underlying learning and memory, requires protein synthesis that occurs primarily during slow-wave sleep. Studies in animal models have shown that the proteins necessary for new synapse formation are produced at significantly higher rates during sleep than during waking rest. Blocking slow-wave sleep after a learning experience prevents the formation of long-term memory traces, even when total sleep time is preserved. Sleep, in this context, is not a passive, consolidation period. It is the biological moment when learning becomes permanent, when what was experienced becomes part of who the brain is. The pineal gland, a small, pine cone-shaped structure deep in the brain, produces melatonin in response to darkness, signaling to every cell in the body that it is night. Melatonin is not primarily a sleep-inducing drug. As it is often described, it is a timing signal, a chemical announcement that coordinates the circadian clocks of peripheral organs with the master clock in the suprachiasmatic nucleus. The liver, the gut, the immune system, the heart, all have their own internal clocks, and all receive melatonin as the synchronizing pulse. Without adequate melatonin signaling, disrupted by artificial light, irregular schedules, or aging, the organ clocks drift out of alignment, running repair programs at suboptimal times. Darkness, it turns out, is a biological instruction. Sleep deprivation accumulates in the body in ways that are not fully reversed by a single recovery night. Researchers studying sleep debt, the cumulative deficit from consistently sleeping less than individual biological need, have found that cognitive and physiological markers of impairment persist for days after returning to normal sleep duration. A 2021 study published in Current Biology found that 2 recovery nights after 5 nights of sleep restriction normalized some cognitive measures, but left inflammatory markers and metabolic function still measurably impaired a week later. The body can recover from short-term sleep loss, but the recovery is not instantaneous. Each night of repair matters, including tonight. Inflammatory markers in the bloodstream rise measurably after a single night of poor sleep. C-reactive protein, interleukin-6, and tumor necrosis factor alpha, all indicators of systemic inflammation, are elevated in sleep-deprived individuals compared to well-rested controls. Even after controlling for other variables, chronic low-grade inflammation is a recognized precursor to cardiovascular disease, type 2 diabetes, and neurodegenerative conditions. Without the cortisol nadir and the T-cell reorganization that sleep provides, inflammatory pathways remain insufficiently regulated. Sleep is one of the most potent anti-inflammatory interventions available, not as a metaphor, but as a measurable biochemical event occurring every night. The gut-brain axis, the bidirectional communication channel between the intestinal nervous system and the brain, is modulated by sleep in both directions. The brain's sleep centers influence gut motility, secretion, and microbial balance. In return, the gut sends signals to the brain through the vagus nerve and hormones that affect sleep quality and depth. Tryptophan, an amino acid found in food, is converted in the gut to serotonin, and from serotonin to melatonin, directly linking gut chemistry to sleep timing. Short-chain fatty acids produced by gut bacteria during their nighttime activity appear to support slow-wave sleep. The digestive system and the sleeping brain are not separate systems. They are in continuous conversation. The lymphatic system, a network of vessels and nodes that drains fluid and removes cellular waste from body tissues, operates at higher efficiency during sleep. Like the glymphatic system in the brain, the body's peripheral lymphatic system increases its clearance rate during rest. Lymph nodes show higher cellular activity during sleep, particularly in immune surveillance and debris processing. The spleen, a lymphatic organ that filters blood and recycles old red blood cells. Also follows a sleep-modulated activity pattern. The body's waste management is not a daytime operation. It is a night shift, running in every tissue simultaneously, coordinated by the same circadian signals that direct the brain's own cleanup. Telomeres, the protective caps at the ends of chromosomes that shorten with each cell division, are maintained by an enzyme called telomerase, which is more active during sleep. Telomere length is considered a biological marker of cellular aging. Shorter telomeres are associated with accelerated aging and increased risk of age-related disease. Studies have found that people who consistently sleep fewer than 6 hours per night have measurably shorter telomeres than those sleeping 7 to 8 hours, even after accounting for other lifestyle factors. Sleep does not stop cellular aging, but it appears to slow the rate of telomere shortening, influencing the biological clock at the level of the chromosome itself. The brain's reward and motivation systems, centered on dopaminergic pathways, linking the ventral tegmental area to the nucleus accumbens and prefrontal cortex, are recalibrated during sleep. Sleep deprivation disrupts dopamine signaling in these circuits, producing blunted baseline motivation alongside heightened sensitivity to immediate rewards. This neurochemical shift explains why tired people often feel both unmotivated for complex goals and unusually drawn to impulsive, high-stimulation choices. The craving for sugar, the shortened attention span, the difficulty sustaining effort. These are not character failures under fatigue. They are symptoms of a disrupted reward circuit that sleep restores to its default calibration. Body temperature drops during sleep onset by roughly 1 to 2 degrees Fahrenheit. And this drop is not a passive byproduct of inactivity. It is an actively regulated process, orchestrated by the hypothalamus, that serves a functional purpose. Many cellular repair processes that occur during sleep are temperature sensitive, operating more efficiently at slightly cooler tissue temperatures. The immune system's inflammatory response, protein synthesis in muscles, and certain aspects of neural repair all show enhanced efficiency when core temperature is lowered. The body's cooling at sleep onset is a preparation of the biological environment for the work that follows. Warm feet, which draw heat from the body's core to the extremities, help facilitate this shift. The liver's circadian clock, its own internal timing mechanism, synchronized with but distinct from the brain's master clock, governs hundreds of metabolic and detoxification reactions. During sleep, the liver increases production of albumin, coagulation factors, and other proteins that support tissue repair throughout the body. It processes and clears circulating cellular debris, damaged proteins, and oxidized lipids accumulated during the day. Alcohol disrupts the liver's circadian clock in ways that persist beyond the night of consumption. One reason that drinking impairs sleep quality even when it initially induces drowsiness. The liver is one of the most sleep-sensitive organs in the body, and its nighttime work serves every other organ's repair agenda. The night is still soft around you, halfway to 40, and your breathing has changed. Slower, deeper, settling into a rhythm the body chose without you asking. Notice the quiet between thoughts. There's no need to hold anything right now. Let each breath carry a little more of the day's weight outward. We'll keep going. Slowly. Bone remodeling. the continuous cycle of bone breakdown and rebuilding that maintains skeletal strength, is closely tied to the sleep cycle. Osteoblasts, the cells that build new bone tissue, are more active during sleep, particularly in the hours following growth hormone release. Osteoclasts, which break down old bone, operate on a more continuous basis. The balance between these two cell types determines bone density over time. Researchers found that chronic sleep restriction is associated with reduced osteoblast activity, lower bone mineral density, and elevated markers of bone resorption, particularly in women. The skeleton, like every other tissue, has a repair window that sleep opens and wakefulness closes. The brain's glial cells, which include astrocytes, oligodendrocytes, and microglia, each perform specific maintenance functions during sleep. In animal studies, oligodendrocytes, which produce the myelin sheath that insulates neural axons and speeds electrical transmission, have been shown to divide more rapidly during sleep than during waking. Microglia, the brain's resident immune cells, increase their surveillance activity during sleep, clearing cellular debris, synaptic remnants, and potential pathogens from brain tissue. Whether the same rate of myelin renewal occurs in humans is still under investigation, but the maintenance role of glial cells during sleep is well supported across species. The sleeping brain is a construction and maintenance site, with multiple cell types performing different jobs on overlapping schedules. The eye's photoreceptors, the rods and cones that detect light, regenerate their visual pigments most effectively during darkness and sleep. Rhodopsin, the pigment in rod cells responsible for low-light vision, is bleached by light exposure and requires a period of darkness for full regeneration. This is partly why prolonged exposure to bright screens in the hours before bed affects not just sleep onset, but visual sensitivity the following day. The retina also flushes its own metabolic waste during sleep, in a process that parallels but is distinct from the brain's glymphatic clearance. The repair of the visual system depends on genuine darkness, not simply closed eyes. The brain's default mode network, which quiets during deep sleep, is not the only network that changes its activity during sleep. The salience network, which monitors the environment for threats and novelty, also shows dramatically reduced activity during non-REM sleep. This reduction allows the body's autonomic nervous system to shift more fully into parasympathetic dominance, the state associated with rest, repair, and recovery. Heart rate variability, a measure of autonomic flexibility and cardiovascular health, peaks during deep sleep. When the parasympathetic system operates with the least interference from the brain's alerting circuits, the nervous system's rest is as real as the body's. Protein folding, the process by which newly synthesized proteins assume their functional three-dimensional shape, is a sleep-sensitive process. Misfolded proteins are a hallmark of several neurodegenerative diseases, including Parkinson's and ALS. The cellular machinery that detects and either refolds or degrades misfolded proteins, the unfolded protein response, and the ubiquitin proteasome system, operates at higher efficiency during sleep. Sleep deprivation increases the burden of misfolded proteins in neural tissue. Researchers have found elevated markers of protein folding stress in the brains of chronically sleep-restricted animals, alongside evidence that sleep restoration partially reverses this burden. The brain's quality control runs most efficiently when the lights are out. The hypothalamus, a small but functionally vast region of the brain that regulates hunger, thirst, temperature, and hormone release, undergoes intensive reorganization during sleep. Its connections to the pituitary gland pulse with releasing hormones throughout the night, driving the sequential release of growth hormone, prolactin, and thyroid-stimulating hormone in phase-specific patterns. The hypothalamic circuits that modulate appetite reset during sleep. Leptin, which signals satiety, rises during sufficient sleep, while ghrelin, which drives hunger, falls. After a poor night of sleep, both signals are dysregulated, resulting in measurably higher caloric intake and preference for high-energy foods that researchers have documented in sleep-restricted participants across multiple studies. The cerebellar circuits that coordinate movement and procedural memory are consolidated during sleep in a process distinct from the hippocampal cortical consolidation that handles declarative memory. Motor skills. The learned sequences involved in playing an instrument, typing, or athletic performance are encoded initially in the motor cortex and gradually transferred to more efficient cerebellar and basal ganglia circuits during sleep. Studies have shown that a night of sleep after learning a new motor task produces measurable gains in speed, accuracy, and smoothness, gains that do not occur with equivalent waking rest. The hands that felt clumsy learning something new become more fluent after sleep, through the brain's offline processing of practice already done. The sleep-dependent consolidation of emotional memory involves the amygdala, the brain's threat detection and emotional intensity center, in a complex dance with the prefrontal cortex and hippocampus. During REM sleep, the amygdala remains active, while its connections to the prefrontal cortex loosen slightly. This configuration allows emotional memories to be reprocessed with reduced suppression, enabling emotional integration, while the near absence of norepinephrine prevents reactivation of the stress response itself. People woken during REM sleep consistently report more emotionally vivid and complex dream content than those woken from other stages, suggesting active emotional work is occurring. Sleep is not emotional numbness. It is emotional recalibration. Arterial walls contain smooth muscle cells that maintain vascular tone, the tension that regulates blood pressure and blood flow. These cells undergo repair and regeneration during sleep, driven by growth factors released under the influence of growth hormone and insulin-like growth factor 1. Endothelial nitric oxide synthase, an enzyme that produces nitric oxide, which dilates blood vessels, shows peak activity during sleep. People who consistently sleep fewer than 6 hours per night show measurably stiffer arteries in their 40s and 50s than those who sleep 7 to 8 hours. A difference that begins accumulating in the smooth muscle repair deficit of earlier decades. Halfway through tonight's journey, the answer to our central question is taking shape. What does the exhausted body do to repair itself while you sleep? It runs a complete, organ by organ, system by system maintenance protocol that waking biology cannot replicate. The brain clears its metabolic waste. The immune system recalibrates. Bones rebuild. Arteries repair. Proteins fold correctly. Memories integrate. Emotions soften. The body that you inhabit is being carefully, methodically restored. Not by anything you are doing, but by something that knows exactly what to do when you finally allow it, the silence it needs. This is what sleep is. Not an absence of life, a different kind of living. You've been listening for a while now. The facts are slower now, settling like sediment in still water. Your body is doing work that your attention doesn't need to supervise. Right now, in this very moment, processes that began at sleep onset are running their quiet protocols. There's nothing to do. Just breathe. Let's drift onward together. The kidneys filter approximately 180 liters of blood per day, producing around 1 to 2 liters of urine. During sleep, this filtration rate decreases, allowing the nephrons, the functional filtering units of the kidney, a reduced demand period during which cellular repair and membrane regeneration can proceed. Aquaporin channels, proteins embedded in kidney tubule cells that regulate water reabsorption, are renewed more actively during sleep. Chronic sleep deprivation has been associated in multiple large studies with accelerated decline in kidney function. Independent of other risk factors, the kidney's quiet night of reduced workload is also a night of careful rebuilding. The choroid plexus, a network of specialized cells lining the brain's ventricles, produces cerebrospinal fluid, and its output increases during sleep. The volume of cerebrospinal fluid cycling through the brain is higher during sleep than during wakefulness, supporting enhanced glymphatic clearance. Recent MRI imaging studies have visualized the pulsatile flow of cerebrospinal fluid through the sleeping brain in real time, confirming that the fluid moves in large, synchronized waves rather than the slow trickle seen during wakefulness. Each slow oscillation of the sleeping brain drives a corresponding surge of fluid inward and outward. A mechanical pump the sleeping brain creates for itself. The brain's energy metabolism during sleep is not simply reduced from waking levels. It is redirected. While the brain's overall metabolic rate is slightly lower during non-REM sleep, certain regions and processes consume significantly more energy during sleep than during waking. The hippocampus, during the replay of daily memories in slow-wave sleep, operates at elevated metabolic rates. REM sleep drives high activity in limbic and associative regions. The brain's energy budget during sleep prioritizes maintenance, consolidation, and repair, shifting resources away from sensory processing toward internal operations that cannot run during the demands of wakefulness. Sleep is not the brain powering down. It is the brain changing what it powers. The enteric nervous system, often called the second brain, is a network of approximately 100 million neurons lining the gastrointestinal tract. During sleep, it follows its own circadian program. Intestinal contractions slow, secretory activity shifts, and the renewal of the gut's epithelial lining, which turns over completely every 3 to 5 days, accelerates. This rapid renewal is one of the body's most intensive maintenance operations, requiring significant energy and protein resources. Sleep provides the reduced demand state in which this renewal can proceed, without competition from the metabolic needs of active digestion. Melatonin is produced not only in the pineal gland but in multiple tissues throughout the body, including the gut, skin, liver, and bone marrow, where it acts as a local antioxidant and repair coordinator. Gut-produced melatonin is estimated to be far greater in quantity than pineal melatonin, though it serves primarily local functions. In the gut, melatonin appears to regulate intestinal motility, protect the mucosal lining from oxidative damage, and modulate the behavior of gut bacteria. The presence of melatonin in nearly every tissue reflects how deeply the repair function of nighttime is embedded in biology. The brain's hippocampus also undergoes structural renewal during sleep. New neurons are generated in the hippocampal dentate gyrus throughout adult life. Though current evidence for this process in humans remains an area of active scientific debate, what sleep research does consistently show is this sleep appears to support the survival and integration of newly formed neural elements, while sleep deprivation reduces their functional incorporation into hippocampal circuits. The cognitive flexibility and pattern separation that healthy hippocampal function provides depend on this ongoing renewal, a process that sleep appears to protect, regardless of the precise mechanisms still being studied. The blood undergoes significant compositional changes during sleep. Platelet activity, which is involved in clotting and inflammation, follows a circadian rhythm with lower activity during sleep. White blood cell counts and the proportions of different immune cell types shift in patterns that reflect the immune system's sleep-dependent reorganization. Natural killer cells, lymphocytes that identify and destroy abnormal cells, show higher activity during the nighttime hours. A well-replicated study found that a single night of sleep restricted to 4 hours reduced natural killer cell activity by 70%. The blood running through the body tonight is doing different work than the blood that ran through it this afternoon. The brain's regulation of pain sensitivity is strongly modulated by sleep. Sleep deprivation lowers pain thresholds, making the brain more sensitive to painful stimuli. Through multiple mechanisms, the descending pain modulation system, which sends inhibitory signals from the brain to the spinal cord to dampen incoming pain signals, is impaired by sleep loss. Endogenous opioid systems, which provide natural pain relief, are less active in sleep-deprived brains. Even partial sleep restriction, sleeping 6 hours instead of 8 for a week, produces measurable decreases in pain tolerance. Conversely, extending sleep in people with chronic pain has been shown to reduce pain intensity meaningfully in some trials. The adrenal glands, small organs perched atop the kidneys, regulate the body's stress response through the release of cortisol and adrenaline. Their rest period comes during the first half of the night. When cortisol reaches its daily nadir, the adrenal cortex renews its cellular machinery and replenishes hormone precursors during sleep. Chronic sleep deprivation keeps the adrenal system in a state of persistent low-level activation, gradually depleting hormonal reserves and sensitizing the stress response. People who consistently sleep poorly often describe a baseline feeling of tension or reactivity that never fully resolves. This is the adrenal system running without its nightly reset. The brain's white matter, the bundles of myelinated axons that connect different brain regions, is maintained during sleep in ways that are only beginning to be understood. Myelin sheaths, which degrade gradually through normal use, are renewed by oligodendrocytes during sleep. Studies have found that white matter integrity is measurably lower in people with chronic sleep disorders than in age-matched controls, and that improving sleep quality is associated with partial recovery of white matter microstructure over months. The pathways that connect thought to emotion, to memory, to action are quietly maintained by the sleeping brain. The brain's thalamus, a central relay hub that processes virtually all sensory information flowing to and from the cortex, plays a key role in generating the sleep oscillations that drive glymphatic clearance and memory consolidation. During non-REM sleep, the thalamus produces rhythmic bursts called sleep spindles, brief oscillations lasting roughly 1 second. Sleep spindles are thought to gate the transfer of information from the hippocampus to the cortex and to protect sleep from disruption by ambient noise. Their density and power correlate with measures of learning and intelligence. The thalamus is not passive during sleep. It is the conductor of the brain's overnight orchestra. The respiratory system undergoes subtle but meaningful changes during sleep. Breathing rate slows during non-REM sleep, reducing the respiratory workload. Airway resistance increases slightly as muscle tone in the upper airway decreases. The normal physiological response to mild hypoxia, reduced oxygen, is somewhat blunted during sleep, which is why sleep apnea is more dangerous than equivalent breathing irregularities during wakefulness. At the tissue level, the cells lining the airways increase their mucus clearance activity during sleep, the cilia beating more actively, clearing debris accumulated during the day. The respiratory system uses the reduced demand of sleep to do its own quiet housekeeping. The relationship between sleep and metabolism extends to the level of individual cells. Mitochondria, the organelles that produce cellular energy, generate reactive oxygen species as a byproduct of energy production. These molecules can damage cellular structures if not neutralized. During sleep, cells upregulate antioxidant defenses and increase mitochondrial repair and renewal. Sleep deprivation increases oxidative stress markers in virtually every tissue studied. The antioxidant reset that occurs during sleep maintains the function of cells that must work at high intensity the following day, including neurons, which are among the most metabolically active and oxidatively stressed cells in the body. The skin's circadian repair program involves the activation of DNA repair enzymes that correct damage accumulated from ultraviolet radiation and environmental oxidants during the day. Nucleotide excision repair, the primary mechanism for correcting UV-induced DNA damage, peaks during the late night and early morning hours, coinciding with the window of deepest sleep, disrupting the circadian timing of this repair process through irregular sleep or chronic light exposure at night. Allows UV-induced DNA damage to accumulate at higher rates in skin cells. The skin's genomic integrity, like its structural repair, is a nighttime project. The brain's cerebral blood flow undergoes distinctive changes across sleep stages. During slow-wave sleep, global cerebral blood flow decreases modestly, consistent with the brain's reduced sensory and motor demands. During REM sleep, blood flow increases dramatically, in some regions surpassing waking levels, supporting the intense neural activity associated with dreaming and emotional processing. These blood flow patterns are coordinated by astrocytes, which regulate local blood vessel dilation based on the metabolic needs of nearby neurons, a process called neurovascular coupling that is itself dependent on healthy sleep for maintenance. The brain's response to the day's emotional experiences during sleep involves a process that researchers call emotional memory reconsolidation. During REM sleep, memories with strong emotional valence are retrieved from storage and then restored in a slightly modified form. This modification typically involves a reduction in the emotional charge attached to the memory, not the loss of the memory's content, but a softening of its affective intensity over successive nights. even significantly distressing memories become more manageable through a biological process of emotional distance-making that the sleeping brain performs automatically. The insulin signaling system, which regulates glucose uptake into cells, is recalibrated during sleep in ways that directly affect metabolic health. During deep sleep, insulin sensitivity increases, meaning cells respond more efficiently to insulin and absorb glucose more readily after a night of poor sleep. Insulin sensitivity in muscle and fat tissue decreases by roughly 25%, a magnitude some researchers compare to the effect of significant weight gain. Chronic sleep restriction is one of the most robust predictors of type 2 diabetes risk in longitudinal studies, independent of diet, exercise, and body weight. The brain's circadian clock, housed in the suprachiasmatic nucleus of the hypothalamus, maintains its timing by detecting light through a specialized photoreceptor in the retina called the intrinsically photosensitive retinal ganglion cell. These cells contain a photopigment called melanopsin, maximally sensitive to short wavelength blue light, the frequency most abundant in sunlight and in LED screen emissions. Exposure to blue spectrum light in the hours before sleep suppresses melatonin production, delays circadian timing, and reduces the depth and duration of slow-wave sleep. The body's internal clock is not stubborn. It is responsive, updating its timing based on the light it receives. The brain generates different types of electrical waves during different sleep stages, and each wave type is associated with specific repair and consolidation functions. Delta waves, slow, high-amplitude waves at roughly 1 Hz, drive lymphatic clearance during deep sleep. Sleep spindles coordinate hippocampal cortical memory transfer. Theta waves at 4 to 8 Hz characterize REM sleep and support emotional processing. Slow oscillations coordinate the timing of all other sleep rhythms. These rhythms are not incidental electrical noise. They are the biological machinery of repair, running in a precise sequence that evolution refined over hundreds of millions of years. The body's peripheral clocks, the circadian mechanisms in every organ and tissue, are synchronized with the master clock in the hypothalamus through hormonal signals, body temperature rhythms, and neural inputs. When sleep aligns with the natural light-dark cycle, these peripheral clocks run in phase with each other, allowing organ systems to coordinate their repair activities. When sleep is shifted, as in jet lag or shift work, peripheral clocks drift out of phase, creating internal desynchrony. Research has found that even 2 days of shifted sleep produces measurable changes in gene expression in peripheral blood cells, affecting pathways related to inflammation, immune function, and metabolism. Among the most intriguing aspects of sleep repair is the phenomenon of synaptic tagging. During learning, synapses that are activated receive a molecular tag, a temporary marking that allows them to be strengthened during subsequent sleep. The tag requires sleep for completion. Tagged synapses that don't receive the consolidation signal during slow-wave sleep lose their tag and fail to undergo long-term potentiation. This process explains why studying and then immediately sleeping produces better memory retention than studying and staying awake. The brain does not just process what happened today. It processes what was marked as worth keeping. The brainstem's reticular activating system, which maintains wakefulness and regulates arousal, undergoes a precisely choreographed withdrawal during sleep onset. Neurons in the locus coeruleus, which produce norepinephrine and maintain alertness, cease firing almost completely during sleep. One of the most dramatic single-cell behavioral changes in the nervous system, the dorsal raphe nucleus, which produces serotonin, also reduces firing during deep sleep. This simultaneous silencing is not a passive fading, it is an active process, driven by sleep-promoting neurons in the ventrolateral preoptic nucleus. Sleep, at the cellular level, is a state the brain constructs deliberately. The brain's capacity for perceptual updating, its ability to revise its model of the world based on new information, is enhanced by sleep. Studies using perceptual learning tasks have found that improvements in the brain's ability to detect fine-grained patterns in sensory data occur primarily during the night, following training, not during practice itself. This enhancement correlates with the density of sleep spindles in the regions processing the trained sensory modality. The sleeping brain is not simply consolidating what it already learned. It is extracting patterns from the day's experience and updating the perceptual model that shapes how tomorrow will be seen. Reactive oxygen species. The oxidative molecules that accumulate as byproducts of cellular metabolism are cleared more effectively during sleep through both enzymatic and non-enzymatic antioxidant systems. The brain is particularly vulnerable to oxidative damage because of its high metabolic rate and relatively modest antioxidant reserves. Melatonin, in addition to its timing functions, acts as a direct antioxidant in neural tissue, neutralizing hydroxyl radicals. The most damaging of oxidative molecules, its production peaks in the early hours of sleep. This antioxidant pulse is one of several mechanisms through which the sleeping brain protects its structural integrity against the oxidative cost of a full day's activity. Somewhere in the terrain of these facts, the answer to our central question has fully arrived. Not as a single discovery, but as a recognition. What does the exhausted body do to repair itself while you sleep? It does this. All of this, simultaneously, in every organ, every cell type, every tissue. The repair is running. Sleep is not the absence of activity. It is the body's most sophisticated repair system, running only when you let it. The exhaustion you feel is not damage without remedy. It is the signal that the remedy is ready. The body has been waiting for you to stop. Tonight, you stopped. Breathe slowly. There's a quiet rhythm running through you right now. The slow pulse of cerebrospinal fluid. The deliberate rhythm of a sleeping heart. The soft oscillation of delta waves in a resting brain. None of it is rushing. Like the deep earth that holds its warmth through winter without any reminder. Patient. Sure. Unhurried. We're almost there now. The vagus nerve, the body's longest cranial nerve, running from the brainstem through the heart, lungs, and gut, is the primary pathway of the parasympathetic nervous system. During sleep, vagal tone increases significantly, promoting rest and repair throughout the body. Heart rate variability, the natural variation in time between heartbeats, rises during deep sleep, a sign of healthy autonomic balance. Sleep is one of the most reliable ways to restore vagal tone. Every night of deep sleep is a contribution to the long-term resilience of the autonomic nervous system. The prefrontal cortex rebuilds its executive reserves during sleep in ways that resting while awake cannot replicate. The neural pathways connecting the prefrontal cortex to subcortical emotional regions which become less regulated under fatigue, producing emotional reactivity and impulsive choices, are restored in their inhibitory strength during sleep. The calm judgment, the measured response, the ability to pause before reacting. These are not traits that some people have and others lack. They are neurological capacities that sleep restores and sleep deprivation erodes. The glymphatic system's efficiency declines with age, which may partly explain the increased risk of neurodegenerative disease in older adults. As the brain ages, the water channels through which cerebrospinal fluid flows become less permeable. Deep sleep, the stage most critical for glymphatic function, decreases in duration and depth across adulthood. Researchers are exploring whether interventions that improve deep sleep in older adults can preserve glymphatic function and reduce the accumulation of proteins associated with Alzheimer's disease. The aging brain is not inevitably declining. It is managing a maintenance challenge that sleep may meaningfully address. The adrenal glands release a small pulse of cortisol approximately 1 hour before natural waking, a phenomenon called the cortisol awakening response. This pulse serves as a biological alarm. It mobilizes energy, sharpens attention, and prepares the immune system and metabolism for the demands of the day ahead. The cortisol awakening response is more robust in people who sleep well and wake refreshed. It is blunted in people experiencing chronic stress and burnout. The morning that awaits is partly built overnight. Sleep promotes the growth and branching of dendritic spines, the tiny protrusions on neurons that receive synaptic input from neighboring cells. During waking learning, dendritic spines grow to form new synaptic connections. During sleep, they are selectively pruned, some are stabilized and strengthened. Others are retracted. This selective process results in a more efficient, less redundant neural network by morning. The brain that wakes after sleep is not merely rested. It has been structurally refined, more selective, more efficient, more precisely tuned to what was learned and what was released. Wound healing is measurably faster when sleep is sufficient. Skin biopsies studied in controlled experiments show higher rates of cellular migration into wound sites and faster epithelial closure in well-rested participants. Growth hormone, released during deep sleep, stimulates the division and migration of fibroblasts, the cells that build connective tissue and close wounds. The body's ability to repair physical damage is not simply a matter of energy availability. It is a sleep-gated process, running most efficiently during the very hours when the body is still. The eye's ciliary muscles, which control the focus of the lens, relax completely during sleep, relieving the sustained tension of a day spent focusing at close range. Intraocular pressure, the fluid pressure inside the eye, follows a circadian rhythm, and the nighttime reduction in pressure is part of the eye's regular maintenance. Elevated intraocular pressure is the primary risk factor for glaucoma. The structures of the eye, like muscles elsewhere in the body, need a period of reduced demand to recover from the sustained work of the day. The body's thermoregulatory system undergoes a circadian shift during sleep. Core body temperature continues to decline for several hours after sleep onset, reaching its lowest point roughly 2 to 4 hours before natural waking. Peripheral skin temperature rises simultaneously as blood vessels in the hands and feet dilate to release heat from the core. This core-to-periphery heat redistribution is coordinated by the hypothalamus and is one of the most reliable biological signals of healthy sleep onset. disrupting this thermoregulatory shift through illness or environmental heat impairs sleep quality at a fundamental level. The hippocampus completes a quiet inventory during sleep. Each day's experiences are temporarily stored as patterns of neural activity. During slow-wave sleep, these patterns are replayed, compressed, abstracted, and forwarded to the cortex for long-term storage. The hippocampus does not simply archive the day, it curates it, forwarding what fits into existing knowledge structures and releasing what holds no further relevance. By morning, the hippocampus is largely cleared, its temporary storage freed for the next day's experiences. Sleep empties the day's inbox and files what matters. The body's connective tissue, the fascia, tendon, and ligaments that hold the musculoskeletal system together, undergoes repair and remodeling during sleep. Collagen, the primary structural protein of connective tissue, is synthesized at higher rates during sleep. Under the influence of growth hormone, the repair of micro-tears in tendon and ligament tissue caused by normal daily movement requires the reduced mechanical loading that sleep provides. The body's structural framework, like its neural architecture, is built and rebuilt during the quiet hours. The brain's slow oscillations, the large, rhythmic waves between neural activity and silence that characterize deep sleep, are generated by the cortex itself. Each slow oscillation has an active phase, called the up state, during which neurons fire in synchrony, and a silent phase, the down state, during which neurons briefly rest. These oscillations are the mechanism through which the brain coordinates simultaneous processes, timing the release of cerebrospinal fluid pulses, synchronizing memory replay, and orchestrating the pruning of synaptic connections, all from the same rhythmic signal. The inner ear undergoes cellular repair during sleep that is not possible during waking activity. The hair cells of the cochlea, delicate sensory cells that translate sound vibrations into neural signals, are among the most damage-prone cells in the body. They cannot regenerate once destroyed, which is why hearing loss from noise exposure is permanent. But existing hair cells undergo membrane repair and ion channel renewal during sleep, restoring their sensitivity and reducing the cumulative effect of daily acoustic load. The rest that sleep gives the auditory system is the active repair of the machinery that hears. The spinal cord's neural pathways are maintained and myelinated during sleep in ways that affect sensory transmission and motor control. Signals traveling between the brain and the body pass through spinal relay stations whose efficiency depends on myelin integrity. In animal studies, chronic sleep restriction has been shown to impair spinal cord myelination, slowing neural conduction and affecting coordination. The body's information highways, the tracts of the spinal cord, appear to be repaired on the same sleep-dependent schedule as the brain's own white matter pathways. The body's lymph nodes, scattered throughout the body and concentrated in the neck, armpits, and groin, increase their filtration and immune activity during sleep. During sleep, lymphatic vessels dilate slightly, increasing flow. Immune cells within the lymph nodes become more active in identifying and destroying pathogens, cellular debris, and abnormal cells. This heightened lymphatic activity during sleep is why swollen lymph nodes during illness often feel most prominent in the morning. The nodes have been working intensively through the night. The immune geography of the body is most active, while the rest of it rests. The heart muscle, the myocardium, undergoes repair during sleep that parallels the repair occurring in skeletal muscle. Cardiac muscle cells have limited regenerative capacity, making maintenance of existing cells particularly important. During the nocturnal dip in blood pressure, the mechanical load on the myocardium decreases, allowing repair processes to proceed under reduced strain. Studies have found that people with sleep disorders affecting nocturnal blood pressure dipping have higher rates of cardiac hypertrophy and left ventricular dysfunction, signs that the heart's nightly repair window is being compromised. The gut's epithelial lining, the single-cell thick barrier separating the gut's interior from the body's circulation, is one of the most rapidly renewing tissues in the body, turning over completely every 3 to 5 days. The renewal process accelerates during sleep. Intestinal stem cells, which generate replacement epithelial cells, are more active during the nighttime hours. In animal studies, sleep deprivation has produced measurable thinning of the gut, epithelial lining, and increased intestinal permeability, allowing bacterial products to enter the bloodstream and trigger systemic inflammation. The gut's wall is rebuilt nightly. Sleep is the shift when construction is fastest. The brain's norepinephrine system, which drives alertness and the stress response, is nearly silent during REM sleep. Norepinephrine is also a vasoconstrictor. It narrows blood vessels, including those in the brain. Its near absence during REM allows cerebral blood vessels to dilate more freely, increasing blood flow to regions engaged in emotional processing and creative consolidation. The temporary withdrawal of the brain's alerting chemistry is not a deficit during REM sleep. It is a condition the brain creates to do a specific kind of work that requires the release of vigilance. The biological clock's reset during sleep is not merely a maintenance of existing timing. It is an active integration of the day's light experience into the circadian system's ongoing calibration. Light detected during the day adjusts the master clock's timing, and sleep consolidates that adjustment. Without sleep, the circadian system's response to light input is less precise. The clock drifts more easily. Regular sleep, at regular times, keeps the body's internal time aligned with the external world. A quiet contributor to metabolic health, immunocompetence, and cognitive resilience across a lifetime. The autonomic nervous system's balance between sympathetic and parasympathetic tone shifts dramatically during sleep. Sympathetic activity, the fight-or-flight branch, decreases during non-REM sleep, particularly during deep sleep. Parasympathetic activity increases, slowing the heart, reducing blood pressure, and enabling the repair-oriented metabolic state throughout the body. Sustained sympathetic dominance, which chronic stress and poor sleep both produce, accelerates vascular aging and increases inflammatory load. Each night of good sleep is a restoration of the autonomic balance that chronic stress disrupts. The body's default state, when allowed to sleep, is one of repair. Every cell in the body contains a nucleus holding the genome, the full instruction set for every protein the cell will ever make. DNA accumulates damage continuously from normal metabolic activity, radiation, and environmental exposures. Cells have multiple DNA repair systems: base excision repair, nucleotide excision repair, double-strand break repair that correct this damage before it can cause mutations. These repair systems are upregulated during sleep. When reduced cellular activity and lowered oxidative stress allow repair enzymes to work with less interference. Sleep is, at the deepest level, a genomic maintenance interval. The glial cells that support the brain's neurons hold their own version of the body's story. They do not fire electrical signals. They do not appear in textbooks as the stars of cognition, but they have been present for every thought, every memory, every moment of the day, maintaining the chemical environment, clearing debris, renewing myelin, and every night, they do what they always do: they make space. They contract, they open the channels, they allow the fluid to flow. The brain's repair is, in many ways, their work. By this point in the night, deep sleep has given way to longer stretches of REM. The brain's activity has shifted from the slow orchestration of repair to the softer, associative processing of experience. Dreams, if they are arriving, are not random noise from a resting system. They are the brain's narrative engine, finding connections between today's events and the accumulated context of a life. The exhaustion that brought you here is being metabolized. The weight is becoming something lighter. The morning that follows sufficient sleep is not merely the end of a period of unconsciousness. It is the arrival of a body and brain that have completed a full maintenance cycle. Glymphatic waste is cleared. Memories have been consolidated. Emotional memories have been softened. Muscles have been repaired. Hormones have been recalibrated. The immune system has been reorganized. The person who wakes is genuinely different from the person who lay down. Biochemically, structurally, measurably restored. This is what sleep gives. Tonight, it is already happening. The science of sleep repair is still young. The glymphatic system was discovered in 2012. The full extent of sleep's role in immune function was clarified only in the past decade. The mechanisms of synaptic homeostasis are still being refined. Every year, new research reveals additional dimensions of what the sleeping body and brain accomplish. The picture that has emerged is one of precise coordination. A biological intelligence running a nightly overhaul so complex that no engineered system has come close to replicating it. Sleep is not a gap in life. It is one of life's most elaborate expressions. The exhausted mind that arrived here tonight was not broken. It was loaded, full of a day's metabolic cost, emotional weight, synaptic overgrowth, and accumulated cellular noise. And sleep is not its opposite. Sleep is its answer. The repair that began the moment you closed your eyes has been running through every fact of this journey. Through every description of a glial cell contracting, and a cerebrospinal wave rising, and a memory softening, and a wound quietly closing. What does the exhausted body do to repair itself while you sleep? It becomes, slowly and without effort, the body that will wake tomorrow, restored, consolidated, and ready. Sleep is not the absence of activity. It is the body's most sophisticated repair system, running only when you let it. And so, the science of repair has brought us gently to its own ending. We traveled from the opening of glymphatic channels in the first minutes of sleep to the soft neural work of late REM, from the cellular chemistry of a contracting astrocyte to the genomic maintenance running quietly in every nucleus of every cell in the body you inhabit. 100 facts. One continuous answer. What you carry now, that you didn't carry when tonight began. Is this the exhaustion was never damage? It was a signal, and you answered it. Sleep is the only laboratory in the world that runs every night, in every body, entirely without instruction, and produces, reliably, the most precise restoration science has ever tried to understand. Breathe gently now. Let your entire body soften into the surface beneath you. The repair work running through you tonight is not waiting for permission. It was already underway the moment you grew still. Let your awareness dissolve into that gentle current, the slow pulse of a system that knows where it's going, into the quiet that was always underneath the thinking, into rest, into the space that sleep creates, and only sleep can fill. But there's no need to do anything now, nothing at all. You've traveled far tonight, through the brain's deepest channels, through the body's quietest work, through 100 facts that were never really about science. They were about you. You can allow these thoughts of sleep and repair to fade now, like the last light leaving a room where something careful has been completed, leaving only the warmth behind. Let your breathing deepen. Let your body feel heavier, as if the bed itself is drawing you gently inward, the way the earth holds everything that rests on it, without effort, without judgment, without end. If sleep comes, let it carry you like a current that was already moving beneath you, smoothly, inevitably, into a place where time and weight no longer matter. If your eyes are still open, the next video is waiting for you, ready to continue the journey. Tomorrow, the body will still be working, the brain will still be at its quiet labor. But tonight belongs to stillness. Rest well, quiet traveler, and remember, the most precise repair system in the known universe has been running inside you, without a single instruction from you, all night long. Good night.